Hello everyone and welcome to AMP2, and this session we are going to cover the urinary system, or the pee maker as I like to call it. So this is chapter 26, and your next exam will cover digestive, metabolism, and this final set of notes, urinary. So a lot of the anatomy of urinary, as we've done all throughout this semester and all throughout AMP1, I'm not really going to go into a lot of the gross anatomical structures of the kidneys and whatnot, because we will be doing that in lab. However, we will talk about the functional aspect of the urinary system. And the kidneys are your functional structures, and they have millions of these little teeny tiny loops that we call nephrons. And the nephrons are kind of the workhorse of the kidneys. They're the ones that do the actual filtration between the filtrate and the interstitial fluid. So just kind of that balancing of water and electrolytes and waste products such as urea and so forth, making sure that in a healthy body that those things that need to be reclaimed are reclaimed, and those things that are true waste products are sent out as waste to go out of the ureters and to the urethra and to the bladder and out of the body. So there are two different types of nephrons that we have. We have what we call cortical nephrons and juxtamedullary nephrons. Your kidney has two aspects to it. You have a medullary aspect to the kidney, which is the middle portion of the kidney, and a cortex of the kidney, which is the outside portion of the kidney. So the cortical nephrons, they're going to be found primarily in that outside portion of it, in the cortex, if you will, of the kidney. The juxtamedullary nephrons are going to have parts of their structures in the cortex, and then the loop, or another parts of it, the nephron will be down into the medulla. So that's what we call it juxtamedullary, because it's juxtapositioned between the cortex and the medulla. So the first part of all nephrons, whether they're cortical nephrons or juxtamedullary nephrons, is the glomerulus. The glomerulus is just a bunch of interlining capillaries where we have the blood that's being brought into the glomerulus through the afferent arteriole, and then you have the blood that's leaving out of the glomerulus in the efferent arteriole. And that blood that leaves through the afferent arteriole is going to go back into main circulation, but the peritubular capillaries that surround the nephron, and they can drain into small venules, and as I said before, return blood back to the venous system. The way that the glomerulus works is it kind of just throws everything it can possibly get out. And then all along the rest of the nephron, there are going to be small fine-tuning adjustments that are made to what we would call the filtrate. The filtrate will eventually be called urine, but right now it's not quite the consistency of urine, so we just call it filtrate. So in the glomerulus, we just kick everything out. The adage I like to use, or the saying, is that the glomerulus is like I am, and it's time to clean. When we're doing spring cleaning, I throw everything out of the garage except for like the cars and the kids' bikes and the lawnmower, the weed whacker, and that kind of stuff. But anything else, old pieces of wood or jump ropes that don't look like they've been played with often, I like to throw it all out. So when it's time for me to spring clean, I want it all gone because I want a fresh slate. So that's kind of how the glomerulus works. It throws everything out. So the glomerulus, the first part of the nephron, is going to be found, well, it's the first part of the nephron, and whether it's a cortical nephron or a juxtamedullary, we have our afferent and efferent arterioles that go afferent going in, efferent leaving out. This big blob purple thingy is the glomerulus. So here we have our cortical nephron, where a majority of it is in the cortex, or the outer portion of the kidney, and then this inner portion of the kidney, the medulla, our juxtamedullary nephron has some portion in the cortex, but then a majority of it is down here in the bottom. Other structures that we'll see, we have our glomerulus, and then the next stop for the filtrate, which is kind of the way to think of it as the pre-urine, is going to be the proximal convoluted tubules. And then after the proximal convoluted tubules, you have the descending loop of Henle, and you have the ascending loop of Henle. And then from there, you go to the distal convoluted tubule, and finally the collecting ducts. So whether you're a cortical or a juxtamedullary nephron, you have all of those same portions, and they have very different different physiological jobs that they do, each portion of the nephron. So once the filtrate reaches a collecting depth, we can now finally call it urine, and the urine will then go down into the renal papilla, where it will be collected into the minor calyx, and then all of the minor calyces will then group into the major calyx, and then they'll go out of the ureters and into the kidneys. So this is where we're looking at, and this whole job here is what we call a renal pyramid. So everything out of that renal pyramid, because there are millions, every time you see a little area that's cut off, that's representing some sort of nephron that's connected there. So that when all of our urine is ready, it goes through the ureter, out of the renal pelvis, and into the bladder. So functional anatomy of a representative nephron, so we're not saying whether it's a cortical or a juxtamedullary, it's just a representative nephron. Parts of the glomerulus we've already talked about. We have the afferent arteriole that's carrying the blood and all of the stuff that's carried in the blood into the glomerulus, and then we have the efferent arteriole, which is carrying the blood back to the rest of the parts of the body. And I want you to pay a little bit of attention to the key that we have here, because this is really a roadmap on what's happening and what's going on along the length of that nephron. So remember, I am the glomerulus. I put everything out. Currently, we have about 5 or 6 bicycles in our garage, and my oldest child is driving now, so I know she's not riding her bike as often as she was, even last year. So we could probably go ahead and get rid of that in the yard sale. So my idea is to, this weekend, throw all of that stuff out. Everything that no one's playing with anymore, basketballs that they're not using, volleyballs they're not using, any of that stuff, let's just go ahead and put it in the garage sale. So I'm the glomerulus. I kick everything out. I kick the baby out with the bathwater, if you will. Only those things that are absolutely necessary get to stay. So for the glomerulus, absolutely necessary things that get to stay: red blood cells, white blood cells, plasma proteins. Pretty much all it's going to keep. Everything else, organic nutrients, ions, water, all of that stuff is going to be kicked out. Follow this green arrow here, and we call it filtrate. So that's what we're calling this filtrate. So now, when we get to the proximal convoluted tubule, the proximal, distal, and the nephron loop are going to be various members in my family, where they're now going to start to, everything that I put out there for the yard sale, they're going to start to slowly reclaim these things and bring them back into the garage, because they absolutely cannot live without them. So for the proximal convoluted tubule, things that are going to be reclaimed are water, ions, and all of those organic nutrients, so things like carbohydrates and proteins and lipids. Those things will be reabsorbed, and when they're reabsorbed, they will go back into the bloodstream. It doesn't show it in this picture, but this entire nephron is going to be surrounded by what we call peritubular capillaries. So there are blood vessels that surround this entire area, so all those things that get reclaimed are going to go back into circulation. We're also going to have some reabsorption of water that takes place in the proximal convoluted tubule. As we go down to the descending loop of Henle, we have primarily water being reabsorbed here, and then in the ascending limb, we're going to have lots of solutes. The primary solutes that are being reabsorbed here are sodium and chloride. So the sodium and chloride that we have being reabsorbed is going to increase the water reabsorption that we have on the other end of the loop. So remember, water follows salt. So the more of this sodium and chloride ions that are reabsorbed, the more water that's going to be reabsorbed on this descending loop. When we get to the distal convoluted tubule, we're going to have the secretion of ions, acids, drugs, and toxins. So we're going to have some things coming back in. So things that were reclaimed that shouldn't have been reclaimed, some ions, some acids, and anytime you use the term secretion in the urinary system, that means it's going to go out of your body. So if you're secreting tears, they're coming out of your body. Secreting sweat, urine, that means it's coming out. So anything that's a secretion means it's going into the filtrate to become urine. So some things, toxins, acids, ions, will get reabsorbed. How much of it is kind of variable depending on what kind of item we're talking about, and it's also variable depending on other aspects of the nephron. We're also going to have reabsorption of some water, sodium ions and calcium ions, and those are all going to be under hormonal control. And then finally, at the point where we get out of the descending, the distal convoluted tubule, then it should be the urine is going to be pretty much the consistency of what we want it to be. So the juxtaglomerular complex is an endocrine structure that's going to secrete two different hormones. It's going to secrete erythropoietin, which we're very familiar with what the job is of erythropoietin, to make red blood cells, and it's going to secrete the hormone renin. And renin's job is to help with regulating blood volume and therefore regulating blood pressure. And it's formed by what we call the macular densa cells and the juxtaglomerular cells. There are 3 organic waste products that we want to find in the urine, and those are urea, creatine, and uric acid. And urea we talked about a little bit in the metabolism chapter, where we said that urea was one of the byproducts or waste products of metabolism of proteins on there. So these products here, we don't have any use for. Your body is, as Fiona Apple would say, an extraordinary machine. It's going to keep those things that are absolutely necessary to keep, and it can recycle them and reuse them in various different ways. If it can't find a use for it, then it is considered to be a legitimate waste. So remember these 3 waste products. So this is just a bigger picture of a kind of the overall picture of urine flow that we saw before, where we had, remember at the glomerulus, we threw the baby out with the bath water, or that's me cleaning out the garage. Everything goes out, except for those things that really need to stay. So in the case of glomerulus, red blood cells, white blood cells, plasma proteins. We are going to have some water to be reabsorbed, and anytime we use the term reabsorption, that means it's going back into circulation. We will have some solutes that are reabsorbed, remember those organic nutrients, carbs, proteins, lipids. Those things are going to be reabsorbed here. We also had the reabsorption of water and different solutes, particularly sodium and chloride solutes on the ascending limb, and that's going to drive the reabsorption of water on the descending limb of the loop of Henle. In your distal convoluted tubule, remember that we had some things that were going back in to become the filtrate. They were going to be the urine, and that included organic waste such as the urea, uric acid, and creatine, various different ions, and we'll talk specifically about which ones get to come back in and how they come back in the next few slides. And then we have some water reabsorption that continues to happen here. And then along the collecting duct is primarily under the control of different hormones, whether it's aldosterone and primarily antidiuretic hormone, and exactly how much water is going to be reabsorbed and how much of a solute is going to be secreted back into the collecting ducts, where we finally have our urine that goes out of the collecting ducts and through the renal patella, into the minor calyx, the major calyx, and then out the renal pelvis of the ureters and into the bladder. So what we define as renal threshold is the plasma concentration at which a specific compound begins to appear. And that changes depending on what the substance is. So the renal threshold for proteins are going to be different than the renal threshold are for plasma. And it's also going to be different depending upon the health of the individual's kidneys. If your kidneys aren't able to filter and reabsorb properly, reclaim things properly, and throw things out that are actual, true waste properly, then the consistency of the urine will not be what it's supposed to be. It's going to be different or changed. And we can make early detections on problems with the kidneys by looking at what's in the urine. We shouldn't have whole red blood cells. That lets us know there's a problem with filtration. We definitely should not have whole white blood cells. We shouldn't have an abundance of glucose. We shouldn't have an abundance of proteins. Anytime we find too much, some of those things in the urine are fine. We expect to see them. But anything that's higher than what we consider to be that renal threshold is an indication of kidney infection, kidney damage, kidney failure. So here, we've kind of been talking about this the entire session, that we've talked about all of these parts of renal function, but we just looked at it from an anatomical perspective, where we looked at where the glomerulus is, kind of talked about what it does, and then the distal convoluted tubule, the nephron loop, ascending and descending, and then the proximal convoluted tubule and the collecting ducts. Now we're going to talk about some specific things that happen on each of these stages of renal function. Now your book lists it as 7 steps of renal function, but you're more than welcome, because this is, let me star this slide because it's super important. So, but you can list these in 5 steps or 4 steps, or I don't care how you do it, bulleted points and make them 15 steps. I don't care, as long as all the information is there. So it's really user's choice on how you want to show me that you understand these steps of renal function. Because as we saw in a lot of our other physiological steps, that one step kind of merges into the next one. So how you separate them or delineate them is totally up to you. So first part, we all recognize is that we have glomerular filtration, where the filtrate, remember filtrate's just a fancy word for pre-pee, not quite urine yet because it's not the right consistency. The filtrate is produced at the renal corpuscle, which is that capsule, or what we call Bowman's capsule as well. So you'll hear it referred to as Bowman capsule, the glomerulus. That filtrate at this point, remember, that's me kicking everything out, it has about the same consistency as blood plasma. So it's kind of like the liquidy portion of the blood without the formed elements, because remember, the glomerulus throws everything out: glucose, proteins, lipids, ions, all of that stuff out. Water, not all the water, but a good chunk of that water is going to get thrown out, and it's up to the proximal convoluted tubule and the remaining portions of the nephron to bring those things back in. So the only thing that stays in circulation at the glomerulus: red blood cells, white blood cells, plasma proteins. So things like albumin, immunoglobulins, and those sorts of things, fibrinogen, those plasma proteins will stay in circulation. Next step we have is the step of the proximal convoluted tubule. So in my family, these would be my children or my husband starting to slowly bring things back in. So it's the active removal of ions and our organic substrates. So all of that stuff that I said was filtrate, or all that stuff that I said, this can go into the, the community yard cell, they slowly start to bring those things in. So there's going to be some ions that are brought back into circulation. So when I say bring things in, we're talking about bringing them back into the body, bringing them back into the blood vessels, into those peritubular capillaries that are surrounding the nephron. We're also going to have organic waste substances can stay in the filtrate, so urea, uric acid, creatine, those can all stay as part of the pre-pee or the pre-urine, but things like glucose, proteins, carbohydrates, water, that stuff is going to be reabsorbed. So as a result of taking the water and taking all of these other entities that we want to keep, like the organic nutrients, carbs, proteins, lipids, we're going to reduce the volume of the filtrate. So it's not going to be nearly as high at the proximal convoluted tubule as it is at the glomerulus. Think about it as my yard cell. When I put stuff out there for the yard cell, there's a ton of stuff out there. By the time at least one of my children or my husband has seen it, they're like, hey, that's a saw that I could use one day, although I don't ever use it. They start to bring things back in. So they're reducing the volume of items I have to sell at my yard cell. Same thing with the proximal convoluted tubule. It's reducing the amount or volume of the filtrate and reclaiming some of those items because they can still be useful to the body. Keep solutions inside and outside the tubule isotonic. So we haven't really generated an osmotic gradient yet, where there's more solids or fluids on the inside or the outside of the tubule itself. It's pretty much isotonic at this point. That will change as we go through the loop. So next step, as we end get to the end of the proximal convoluted tubule, and we go to the descending limb of the loop of Henle, or the nephron loop. Water is going to move into those peritubular fluids, and what we have here is highly concentrated tubular fluid. So now that we've had more water is leaving out of the tube, and it's going into the blood vessels surrounding the tubes, those peritubular capillaries, what's left to be in the tube as the filtrate is now more concentrated. Think about it like this. If you took all of the water out of a juice, and it's really, if you took the water parts out of it, then you're left with like the crystals of the sugar or whatever else that's in there, so it becomes much more concentrated. Same thing with detergents. When they say, oh, we have the same cleaning power as we did before, but it's just more concentrated. All they did was just take the water out of it for the most part. There's just not as much water put into the detergent when they make it. So as a result, it's more concentrated and it's in a smaller bundle. Same sort of thing here. As water moves out of the filtrate, and then we're going to have a higher concentration. Now on the thick ascending limb, we're going to actively transport sodium and chloride out of that nephron tube. So as we have transported the sodium and chloride out of the nephron tube, on the other side, on that descending limb, that's driving that water to leave out. So before, in this step here, we just talked about how water moves into the peritubular fluid, leaving highly concentrated tubular fluid. Okay, but we didn't say how. How does that happen? Well, at the ascending limb, we have special transport systems that are actively taking sodium and chloride out. NaCl, last time I checked, was just the regular molecular formula for table salt. So as that salt has gone out of the ascending limb, that concentration of being higher concentrated in salt and higher solutes on the outside of the nephron tubes has driven water to want to follow it. So we've now created an osmotic gradient on this ascending limb. The urea accounts for a higher proportion of the total osmotic concentration. So inside the loop itself, we have a very high urea concentration, which we already started kicking out away from the glomerulus and the proximal convoluted tubule and so forth. But at this point here, we've kicked out sodium and chloride, which we can use for various things in your body. You're very familiar with how sodium is used for synaptic transmissions and muscle contractions, and then how chloride is used for the alkali tide or the chloride shift in both your respiratory system as well as in your digestive system. So these two ions, we know that we have a total use for them in the body, so it makes sense that they're going to be reclaimed. When we get to the distal convoluted tubules and the collecting ducts, so we're out of the loop of Henle or the nephron loop, we're going to make final adjustments. So there are going to be some things that are brought in or secreted from the peritubular fluid or the interstitial fluid or from those blood vessels, and some things that are going to go back out that are going to be reabsorbed back into the circulatory system. The concentration of the osmotic gradient is adjusted through active transport. So once again, as I said before, this reabsorption and secretion is happening, and how much of it, since these are just final adjustments, it's variable. When we get to the collecting ducts, more readjustment that's taking place, but at the collecting ducts, a very important thing that's happening here is that we now are under the control of antidiuretic hormone, which is going to determine the final urine concentration. And with antidiuretic hormone, this is one that we've talked about in the blood vessel chapter, or in the blood chapter early this semester, and we also talked about it at the end of AMP1 in the endocrine chapter. So a diuretic is something that makes you want to have to go use the bathroom, makes you want to go pee. But an antidiuretic will be doing the opposite of that. So it's going to retain water. So the more antidiuretic that is released from your pituitary gland, the more concentrated your urine is going to be, because it means it's retaining that water. The less antidiuretic hormone that you have, the higher the volume of urine you're going to have, and the more dilute it's going to be, because it's going to be a large concentration of water. And then the vasa recta, step 7, is just absorbs those solutes and water that have been reabsorbed by the nephron loop in the ducts, and just helps to maintain the concentration gradient of what it should be in that middle portion of the kidney we call the medulla. So urine production ends once this fluid that we've been calling filtrate enters into the renal pelvis. And all of that filtrate, now that we can call urine, goes out of the renal pelvis, through the ureters, down into the bladder to be stored until it is evacuated out of the body through the urethra. So some terms that we've kind of been using quite frequently, just so that you can have a visual or something to make use your for your flashcards. Reabsorption is going to be the recover of useful materials. So whenever we talk about something being reabsorbed, that means we're putting it back into circulation. It's useful. Secretion, on the other hand, means it's going to be the ejection of things that are not useful. So waste products, toxins, any solute that we either have enough of or we have too much of. So hydrogen ions, because our pH of our blood likes to be slightly basic. So excess hydrogen ions, we're going to get rid of those as well. All along the length of the nephron, you're going to have reabsorption and secretion that takes place. And we've already talked about some of those changes that happen from one segment, aka the glomerulus, to the proximal convoluted tubule, to the nephron loop, to the distal convoluted tubule, and the collecting duct. There are some fine changes that take place on what gets reabsorbed and what gets secreted. Sodium ion reabsorption is pretty important in every step of the proximal convoluted tubule. So that very first section of tubes after you leave the glomerulus. Ions are going to enter tubular cells because there are leak channels in those cells that make up the proximal convoluted tubule of the nephron, and they will just leak out. There are also sodium late co-transport of organic solutes. So there are different organic compounds that when we say a co-transport, that means that they are going to move out together. So things like glucose and sodium can both move out together. And then a counter-transport system, remember how I said before in the previous slide that you don't want too many hydrogen ions to be in circulation. You don't want too few, but definitely not too many, because what happens there is that your pH will get lower than 7.4, and we like for our pH to be slightly basic at 7.4. So the counter-transport means that we'll take in sodium ions and kick out hydrogen ions. So we're going to reclaim those sodium ions, and then the hydrogen ions are going to become part of the filtrate. So these are just various ways in which we are able to reabsorb sodium ions in the proximal convoluted tubule. And remember, reabsorption means that we're putting it back into circulation. And here's a pretty picture of all this happening. So I'm going to, before I even really talk about it, I'm going to let you guys kind of chew on this and look at this and kind of digest it and see what you find here, see what your understanding is after we've talked about all of these things previously. So just kind of have a moment to just go over this slide. All right, so remember in the previous slide that we talked about how there are those different ways that we could reabsorb those sodium ions and put them back into circulation? And one of them was this co-transport mechanism where we'd bring in an organic nutrient such as glucose, and then along with that we'd bring in a sodium ion. So there is that process happening. We also have these leak channels where some of the sodium in that tubular fluid, when we say tubular fluid here, we're talking about the filtrate. Notice it has kind of a yellowish color because it's going to eventually become urine. We can have sodium leak in. We can also have that third mechanism of getting sodium ions in the proximal convoluted tubule back into circulation, and that was through that counter-transport mechanism where we would take hydrogen ions out of circulation and then put them in the tubular fluid, and when we exchange the hydrogen ions, we swap them in for sodium ions. So we have that happening there as well. So these things on the blue arrows are going to be items that we reclaim, and we want to keep those in circulation. We have a sodium-potassium pump that you all are very familiar with from the nervous system, where for every 3 sodium that are going in one direction, 2 potassium will go in the other. We want to reclaim more sodium than we reclaim potassium because sodium has a I won't say it's more important, but it has a more robust role than potassium does in the body. So the sodium ions, we want to reclaim those more. Hydrogen ions, remember, we don't want to keep as much of them because it can make our pH go down too low, and we want our pH to stay slightly basic. So it makes sense that they can leak through the cells of the proximal convoluted tubule and then be used as an ion needed for countertransport. So on to the thin descending limb, very permeable to water. It's relatively impermeable to solids. What we're really saying here is that mostly water comes out of the descending limb. We don't have a whole lot of solute particles or ions or organic nutrients and things like that going in and out of circulation here. We're just talking about pulling water out of the nephron loop and putting it back into circulation. On the ascending limb of the loop, however, it's a little bit different. It's contains it's relatively impermeable to water and other solids. So without having this active transport mechanism, it wouldn't be able to push things like sodium and chloride out. So if we just left it alone and it didn't have this active transport mechanism, then it would kind of behave in the same way as the thin limb. However, it does have mechanisms and it does have proteins that allow for sodium and chloride to be pumped out of what's called the filtrate into the peritubular fluid. So we can pump those out. And as a result, because we can pump out sodium and chloride, water is going to follow from the descending limb. So sodium and chloride pumps that we find on those that ascending limb of the loop of Henle or the nephron loop, they're going to elevate the osmotic pressure or concentration of the peritubular fluid. More solutes than solvents, more salt than water outside the tubule. So that's going to drive the water that's in the tubule out of the tubule. That's all that's saying. It's going to cause the osmotic flow of water out of the thin descending loop into the peritubular fluid, and it will increase the solute concentration in the thin descending loop. So not as much water is in that ascending loop because it's all been pulled out by the descending loop, which really is just a fancy way of saying all of this because we've said this several times before. So we have another picture here. This is looking at the countercurrent multiplication system and how that has an effect on the urine. And then remember that we have these pumps that will actively transport sodium and chloride into the peritubular space so that water can follow on the descending limb. So where we have in our tubular fluid, we have things like urea, and we definitely want to keep that as part of the filtrate because it is an organic waste product. Chloride ions, along with potassium and sodium, can come in through what we call co-transport. As they come in, the chloride and the potassium come in on another co-transport system, and then the sodium and the sodium is brought in because that potassium is going to go back out as a result of the sodium-potassium exchange pump, 3 for 2. Remember how that process works? So with those 2 potassium ions that came into the peritubular fluid, since we really don't need as high a concentration of them, we can go ahead and use those as our countertransport mechanism to pump them back into the urea. And not the urea, the urine. And then here's a picture that's looking at how the sodium and the chloride concentration in that peritubular fluid is driving water out. So we have how concentrated the urine is. It's what these numbers are dictating here, and how concentrated the urine is and how much we have a lot of water that flows out, and it flows out as a result of the sodium and the chloride that's being actively pumped out, as we had on this picture here, to allow for that water, that exchange to take place. So once again, I'll give you a chance to just kind of look over this slide and get some meaning from it. So remember, we have the concentration of the sodium and the chloride ions in the peritubular space in that medulla is driving the water out. And so we have a lot more concentrated filtrate at this point here. And then the distal convoluted tubule and the collecting ducts, they're impermeable to urea, so it's going to stay in the filtrate itself. But how much water comes out, it's variable. And remember, the reason that that's variable because it's in part is because it has the collecting ducts are under the control of antidiuretic hormone. So aldosterone is a hormone that's produced in the adrenal cortex, or the outside portion of the adrenal gland, or the suprarenal gland. Sometimes your text refers to it as that. That's those little hats that sit on the kidneys. It controls those ion pumps and channels, and a big job of it is that it's going to stimulate the synthesis and incorporation of sodium pumps and channels, so that in the plasma membranes along the distal convoluted tubule and the collecting ducts, we have these pumps that are come out. So the big job, and kind of how we refer to it when we were in ANP1, is that aldosterone's job is to reduce sodium loss in the urine. So as a result of reducing sodium loss in the urine, it's also going to, by default, reduce water loss in the urine. But remember, final adjustments are made with how much water is in your urine by another hormone called antidiuretic, or ADH. Hydrogen ion secretion, and remember, if we're secreting it, we are not keeping it in circulation. We're putting it back into the filtrate. We want to get rid of it. That's what makes your urine acidic. So it acidifies that tubular fluid, or the contents of the fluid in your nephron, which is your filtrate or your pre-pee. If we have too many hydrogen ions in circulation, they can elevate your blood pH. How the amount of hydrogen ions that are secreted and put into the filtrate is going to accelerate when your blood pH gets too acidic. So when your blood pH is too acidic, your body is going to allow for more of that to be in a healthy set of kidneys, in a healthy body, for more of it to be filtered out in your urine, because we want to keep our pH slightly basic. So here we have some bicarbonate, which is a buffer, being reabsorbed back in. And some of this you've kind of already seen, so I'm going to kind of talk about it a little bit and then let it sort of digest for you in a second. So any carbon dioxide that we have can kind of just diffuse its way through those peritubular cells of the distal convoluted tubule. And if it hooks up with water, it's going to make this carbonic acid, which you're very familiar with. Then it can dissociate under carbonic anhydrase, that enzyme, to bicarbonate and hydrogen ions. The hydrogen ions we want to put out. It's cool that we're putting that guy out because A, we don't want it in circulation, it'll make our pH too low, and B, we can use it as a counter-transport or exchange pump, so we can bring in something we do want, sodium, in place of it. And so that sodium just comes back in and goes back into circulation. Hydrogen ions can also be moved out through the same type of exchange pump that we see here and put into the tubular fluid. The bicarbonate, we like bicarbonate as a buffer because it's HCO3 minus, which means that if there are any excess hydrogen ions that are in circulation in your body, it can do a really good job tying them up. Hydrogen ions and bicarbonate ions we're going to talk about a lot when we get to our next chapter of electrolyte balance and water balance. And then we have some bicarbonates we will lose out, but we don't lose them out as bicarbonates. They go through a process of amino acid deamination, so the amino group will be lost out. And once again, we can lose out that amino group and bring back sodium in its place. Chloride ions, we can exchange the bicarbonate for that chloride ion, and it can go out as well. And then what we end up here is we have ammonia chloride, which kind of gives this distinctive smell of urine, because believe it or not, urine that leaves out of a healthy body that is free of a urinary tract, bladder, or kidney infection is sterile. So that smell that you get from urine, that characteristic urine smell, if you will, is from the ammonia chloride. Now, once the urine touches any part of your body or touches the toilet, it's no longer sterile because the bacteria that are hanging out in your body have now contaminated it. But in a healthy body, free of urinary tract infections or kidney infections, should be sterile. So acidosis, two different types we'll talk about. Ketoacidosis, which we talked about a little bit at the end of the metabolism chapter. It develops in cases of starvation or diabetes mellitus. And what's happening there with ketoacidosis is that you have a lot of these ketone bodies that are formed because your body, instead of breaking down lipids and carbs, which are its two favorite things to get energy out of, it's going to start breaking down proteins. So in severe starvation, the body will start to break down its own proteins. And if you have diabetes, that's not managed very well because you don't have enough glucose. Glucose is the problem, not not having enough of it. Because you don't have enough insulin to allow glucose into your cells, then you will have your cells are literally starving, so they'll start to break down proteins and can develop into this ketoacidosis. So when they talk about a diabetic coma, it's actually as a result of having your body break down these proteins because you just don't have enough insulin. So that's kind of what can put you in a diabetic coma. Lactic acidosis. Acidosis happens to all of us, whether you're diabetic or, usually for folks that are starving, they don't really have this issue. But that's just after exhaustive muscle activity. And your body does have mechanisms to get rid of that lactic acid buildup through the Cori cycle in your liver. Alkalosis is the opposite end of this. This is where we have abnormally high blood pH, so that instead of it being lower than 7.4, it's higher than 7.4. It can be caused by prolonged aldosterone stimulation. And remember, aldosterone's job is to maintain those sodium ions, to keep more of those sodium ions in there, and by which more water ions are going to follow, but then it can also alter your blood pH and make it too high. So the way that your body responds to acidosis is that the proximal convoluted and distal convoluted tubule will start to take off the amino groups or deaminate the amino acids in order for those excess hydrogen ions that are floating around to be tied up. So we're going to have lots of ammonia ions and bicarbonate ions as a result of this. The ammonia ions we'll get rid of in the tubular fluid, or we can get rid of them in the filtrate or the urine or the pre-T. The bicarbonate ions, we want to put those into the bloodstream so that they can tie up any of those excess hydrogen ions to bring the pH back up to where it needs to be at 7.4. Remember that your water and solute loss in the collecting duct is primarily under hormonal control, chiefly aldosterone, which controls sodium ion pumps. The action of aldosterone is opposed by natriuretic peptides. The brain and the atrionatriuretic peptides will cause the aldosterone production to stop. And then antidiuretic hormone controls water permeability, and it can be suppressed or opposed or stopped by natriuretic peptides as well. You have two different types of water absorption that happens at this, chiefly at the collecting ducts in the distal convoluted tubule, obligatory and facultative. Obligatory water result usually recovers about 85% of the filtrate. This is where the water movement cannot be prevented because it's just as a result of osmosis. Facultative water reabsorption is where water is reabsorbed along the collecting system ducts in the distal convoluted tubule, because how much of it is reabsorbed is as a result of how much antidiuretic hormone this area has been exposed to, or how much of it has been released. So antidiuretic hormone, the way that it works is that it causes aquaporins to appeal in the cell membranes, so it increases the rate of water absorption or water movement. The more antidiuretic hormone you have released, the more number of water channels that are going to be produced, and the water permeability of the distal convoluted and the collecting ducts is going to increase. So the more antidiuretic hormone you have, the more concentrated and the lower the volume of urine you're going to have. The less antidiuretic hormone, the opposite. So this is a picture that we've kind of seen a couple of times before. Basically, the only thing that's changed is that we're looking at antidiuretic hormone. So when we have, this is the effects of antidiuretic hormone, when we have it present, when it's not present, we have large volumes of very dilute urine because we're not reclaiming as much water. Notice that there's no water reabsorption happening along here. Whereas in the presence of antidiuretic hormone, we have lots of water reabsorption happening, so there's a smaller volume of concentrated urine. Remember that it is, and I hate when your book does this, but it's actually your pituitary gland that's connected to your hypothalamus that releases regulating hormones that secretes antidiuretic hormones. So your hypothalamus is telling the pituitary gland to continuously secrete low levels of antidiuretic hormone, not the hypothalamus directly. At normal antidiuretic hormone levels, the collecting system reabsorbs about 17 liters per day, or 9.3% of the filtrate. Diuresis is what we consider the elimination of the urine. Typically indicates production of large volumes of urine, but just diuresis itself is just the evacuation of urine from the body. Diuretics are drugs that promote water loss in the urine, and we can use diuretic therapy to reduce blood volume, which will therefore reduce blood pressure, and also in cases of edema or swelling, we can reduce that by administering diuretics so that we can get rid of some of that extracellular fluid volume. So a little bit of blood acid-base balance here. We've talked about this a little bit already, and we talked about how too many hydrogen ions can be a problem, brings the pH of the blood down too low. It could cause all sorts of issues. So we have bicarbonate ions that help to tie that up, and we have ammonia ions that help to tie up those excess hydrogen ions. And ammonia would go into the filtrate, and bicarbonate just kind of hangs around floating through the circulatory system. Not only have we talked about it in this urinary chapter, but we've also talked about it in the respiratory chapter where we talked about the partial pressure of carbon dioxide. So anytime we have increases in the partial pressure of carbon dioxide, it's going to decrease our pH, so it's going to make it too low. So not only does respiratory have a role in this, but so does urinary. Anytime we have increases in the bicarbonate ions, that's going to increase the pH. So we have more of these bicarbonate ions that are going to tie up those excess hydrogen ions, so there won't be as many of them to make the blood as acidic, so the pH is going to be higher. Normal pH range is 7.3 to 7.45. We don't want to go any higher than that. Anything lower than that is acidosis. Anything higher than that is alkalosis. So low pHs can depress the central nervous system, cause coma, and even death. High pHs can cause an overexcitation of the central nervous system, the muscles to experience tetany, or kind of to just have these sustained contractions until they just can't hold it anymore and they completely give out, extreme nervousness, and convulsions, or respiratory arrest. So either one of these, acidosis or alkalosis, is not a good thing. So the causes of alkalosis and acidosis can be either respiratory in nature or metabolic. So the pH imbalance, and I'm starring all of this or sunning all of it because these are great options for short answer questions and great options for fill in the blank, primarily short answer question type things. So respiratory acidosis or alkalosis, that's respiratory in nature, is a pH imbalance that's due to the respiratory system failing. And so the partial pressure of carbon dioxide is not within its normal range. Normally, the partial pressure of CO2 needs to be between 35 and 45 millimeters of mercury. Anytime it's out of that, then we can have acidosis or alkalosis taking place. If we have acidosis or alkalosis taking place and it is metabolic in nature, that means that the problem is not with the respiratory system, it's with the amount of bicarbonate ions, that they're out of balance. Either they're too low or they're too high. And here's the range for bicarbonates, between 22 and 26 molar equivalents per liter. So for respiratory acidosis, possible causes are anything that can compromise the exchange of gases or lung ventilation, so emphysema or cystic fibrosis, so that you're not exhaling as much of the carbon dioxide off as you need to. Your respiratory centers can be depressed as a result of drug use, such as barbiturates, that will kind of depress your respirations and makes them very slow to take place, so that you're not, once again, getting rid of as much CO2 as you need to. And shallow breathing. So once again, all of these causes here are relating to the same thing. You're not blowing off enough CO2. So as a result, you're going to have a buildup of hydrogen ions. So what happens with your blood pH is that it's going to become less than 7.45. And then you have your partial pressure of carbon dioxide is greater than the higher end of that normal range. So the partial pressure of carbon dioxide should be between 35 and 45 millimeters of mercury. Anything greater than that, and we have a low pH, tells us that we have acidosis taking place, and it's respiratory in nature. Respiratory alkalosis, hyperventilating, blowing off too much of that CO2, so we're not having as much of those hydrogen ions. In asthma, pneumonia, or anxiety, if the pH is higher than 7.45, but our respirations here are within or lower than 35 millimeters of mercury, then we say that yes, this is alkalosis, and it's respiratory in nature. Metabolic causes of acidosis, getting rid of your bicarbonates, severe diarrhea, renal disease, your kidneys not getting rid of metabolic acids like we want to, ketoacidosis, starvation, we talked about that, having all these excess hydrogen ions as a result of breakdown of proteins, excessive alcohol ingestion results in excess acids in the blood, the extracellular concentration of sodium ions is happening more instead of hydrogen ions. What we have here is that your pH is lower than 7.35. Problem, if we see that the respiratory, the carbon dioxide concentration is within its normal parameters of 35 to 45 millimeters of mercury, then we check our bicarbonate levels. And if our bicarbonate levels are higher than 22 molar equivalents, it's on the high side of that, then what we have here is that there are too many hydrogen ions being tied up because there's too many bicarbonate ions, and the cause of this acidosis is metabolic in nature. Metabolic alkalosis, getting rid of too much hydrochloric acid by vomiting. Diuretics cause a low sodium concentration, ingestion of too many acids, constipation, prolonged retention of feces results in lots of bicarbonates being reabsorbed and put into the fecal matter, and we want to keep them in circulation. So your pH would be higher than 7.45, and also it would be higher than normal as the amount of bicarbonates that we've reabsorbed. So we're tying up too many hydrogen ions, and it's causing your pH to be too high. And we say that what's causing it is not the problem with your respiratory system, it's a problem with the bicarbonate ions, or it's metabolic in nature. So typically what happens in a healthy body is that when one system is failing, the other is going to try to compensate for that, and we call that compensation. So the rules that we use to determine whether acidosis or alkalosis is respiratory or metabolic in nature, or there's compensation that's taking place, is that we look at first the pH. Is the pH within the normal range? If it's higher than 7.45, it's alkalosis. If it's lower than that, then it's acidosis. The next thing that we check is the carbon dioxide levels, so that if the pH is acidic and your carbon dioxide levels are higher than that upper range, between 35 and 45 is normal for carbon dioxide, then we say that yes, it is acidosis because your pH is acidic, and it's respiratory acidosis. If we have the case that the pH of the blood is too acidic and the carbon concentration of the carbon dioxide concentration or the partial pressure carbon dioxide is below 35 millimeters of mercury, we say, well, it's not really a problem with the respiratory system, and has to be metabolic. But your respirations are, the concentration of CO2 is lower than what we need it to be. You have more hyperventilation that's taking place because we're trying to compensate for that. So we have metabolic acidosis with respiratory compensation. If the partial pressure of carbon dioxide is normal and we still are acidic, then we say it's metabolic acidosis, and then there's no respiratory concentration or compensation. And then the next thing that we would do is we would check these bicarbonate levels. So as a sample question here, we have a pH that's 7.45, and the carbon dioxide concentration is 45 millimeters of mercury. So would this be respiratory, or first of all, is it acidosis or alkalosis that we're talking about here? Well, it's higher than 7.45, so it's on the alkaline side of things, it's alkalosis. Now, is it respiratory in nature, or is it metabolic in nature? Do we have a problem with our CO2 levels? Nope, CO2 levels are in the normal range, so it has to be metabolic in nature. And because the CO2 levels are in the normal range, we can't even say that we have respiratory compensation taking place. So our next sample, pH is 7.32. Is that acidic or is that alkaline? It's lower than 7.35, so it's acidic. And then our partial pressure of carbon dioxide is 51. So we have a very high partial pressure of carbon dioxide that's taking place here. So since that's not in its normal range, then it either is going to be respiratory in nature, or we're going to have it metabolic in nature, and we have compensation that's taking place. So let's go back and look at this one. So if we decided that our pH was acidic, and that we have the partial pressure of carbon dioxide, in our example, was at 51. It was not within our normal range, so it's over 45, so it has to be respiratory acidosis. If I had made that number smaller, if I had made this like 25 or 30, then we could say it was metabolic in nature, and it was, and we have respiratory compensation. But remember that when you have respiratory acidosis here, what's happening with your respiratory system is that you are holding on to too much of this CO2, so you're not blowing off those excess carbon dioxide and blowing off those hydrogen ions. So you're holding on to too much of it. So if you're holding on to too much of it, it could cause your pH to become acidic. Now, when we have that compensation taking place, that means that our pH is acidic, and then the partial pressure of carbon dioxide is lower than what we want it to be. It's lower than 35. So what that means is that your body is kind of hyperventilating to get rid of some of those excess hydrogen ions through getting rid of that carbon dioxide in order to compensate for the fact that your metabolic systems are not regulating the hydrogen ion concentration. All right, so that is the end of the urinary chapter. Next one that we'll go over are water and electrolyte balance in our very next section. Have a great day.